So welcome everyone. Welcome to this EUFORGEN webinar series in collaboration with the Horizon Europe OptFORESTS project. So today we are going to deal with the genetic decline, but I will give the floor to Marimete to properly introduce this webinar. Marimete, the floor is yours.
Thank you, Anna Maria. Yeah, welcome to the joint OptFORESTS EUFORGEN webinar. So my name is Marimete-Yanes. I'm a researcher working with forest and genetics at the Norwegian Institute of Bioeconomy Research and one of the participants in OptFOREST. So OptFOREST is a European project led by INRAE in France. And its main goal is to support conservation and sustainable use of forest genetic resources and thereby increasing the adaptive capacity of European forests in the face of climate change and other environmental and societal challenges. So within the project, one of the activity focuses on evolution-oriented management of declining forest and restoration efforts, including evaluation of need of genetic enrichment and development of evolutionary management strategies. And working with these topics, we saw the benefits of bringing together more cross-disciplinary perspective on how to detect genetic decline in natural populations, combining theoretical and practical perspectives with case studies from different groups of organisms. And today we have 3 speakers representing the nature conservation community. And the main goal of the session today is to explore measures, indicators, maybe thresholds of decline, and discuss how and when management intervention might be warranted. So in order to safeguard genetic diversity. Tomorrow, there will also be a session, and the focus tomorrow will be topics from forestry genetic community, focusing on evaluating genetic change in forest stands, practice of breeding, and ex situ conservation strategies. So this means the OptFOREST group has developed this webinar program. He's very happy that the invited speakers have accepted the invitation. So thank you very much. And we are also very happy for you participants joining the webinar. And the first speaker today is Alicia Mastretta-Yanes, and she's an evolutionary biologist and conservation geneticist working as a senior researcher leader at the Royal Botanic Gardens in Kew. And you see her title of the talk on the slide here. And the second speaker is Geir Bolstad, an evolutionary biologist and population geneticist working as a senior researcher at the Norwegian Institute for Nature Research. And the third speaker is Ophélie Ronce, also evolutionary biologist studying how evolutionary processes influence biodiversity adaptation to global change. She's a senior researcher and research director at the Institute of Evolutionary Science of Montpellier. So we will have a discussion afterwards that's going to be led by François Lefèvre from INRAE Avignon, and François is also the leader of the work package Forest Management and Genetic Diversity in OptFOREST. So, Anna Maria, then it's you.
Thank you very much. So we will start with our first speaker, and then we will have a 5-minute Q&A after each speaker. So just these Q&A questions will be very fast, just for some clarifications. So keep your main questions for the discussion at the end of the webinar. So I'm giving the floor now to Alicia. And Alicia, please, the floor is yours.
Thank you. Just testing if you can hear me and if you can see my presentation as shared.
Yes, yes.
Good. We are starting with an achievement. Well, thank you to the organizers for this webinar. I think it's very important, and I am very excited to be talking here today. The title of my talk is How to Detect Genetic Decline Using Genetic Diversity Indicators from National Reporting to Local Management. And I hope that this will open the door for a very nice discussion at the end. The key takeaways that I want you to have from this talk is that genetic diversity indicators are feasible at the scale for many species, leveraging existing data and efforts that are already ongoing. I also want you to take away that many species populations are sadly losing genetic diversity, but that there are options to stop this because we are in the right moment to do so. There are improvements for the methods that I'm going to mention that are needed, but there are very good opportunities for doing so. And I think that this network is part of the opportunities that will be used. And then I want you to have this, at the end, sense that policy and management impacts are feasible for genetic diversity. So I want to start with something that we probably all share, and is that we just love genetic diversity because it's part of the beauty of nature. Sometimes we even call different genetic variants with different words like panther and jaguar, which is just an allelic variation. We also know that to have genetic diversity is to have options for adaptation and therefore for ecosystem resilience. And this is why your whole network is based on. And finally, also genetic diversity is part of the different flavors we eat, like different colors of maize tortillas or chiles, which are from where I am from in Mexico. And yet, genetic diversity is largely neglected by conservation policy and by management. And as an example of that, I want to focus on the highest level international policy on biodiversity, which is the Convention of Biological Diversity. This is a multinational agreement signed by basically all of the countries in the world that has three key objectives: the conservation of biological diversity, the sustainable use of the components of biological diversity, and the fair and equitable sharing of the benefits arising from using genetic resources. It was signed in 1992 in Rio, Brazil. And this is the moment where the term biodiversity or biological diversity was formally starting to be used. And I want you to highlight that genetic diversity or diversity within a species is recognized since 1992 as part of the Convention of Biological Diversity. However, it has not been taken into account as well as species and ecosystems. And then in 2022, a key milestone for the CBD happened, which was the adoption of the Kunming-Montreal Global Biodiversity Framework. What makes different this framework is that it includes not only good wishes, but also specific goals, targets, and indicators to be able to monitor if those goals and targets are being fulfilled. This was adopted at COP15, and it's a major achievement. It's like the climate COP of Paris, but for biodiversity. Because the Global Biodiversity Framework has a monitoring framework, it therefore needs indicators. And these indicators are imperfect because the policy is also imperfect, but they have to balance a scientific and the political realities of being able to track biodiversity. They have to be simple and easy to explain because they have to be used by all sorts of people. They have to be also feasible to measure and to be affordable at the scale in all kinds of countries, not only in European settings or in African settings or in Latin American settings, but all over the place. The first report is due in February 2026, and the next one is going to be in 2029. The Global Biodiversity Framework is also very important because for the first time, it includes genetic diversity with the same level of importance that species and ecosystems. Genetic diversity is mentioned in Goal A and Target 4, referring to the maintain and restore the genetic diversity within and between populations of all sorts of species, including wild and domesticated. But you may be thinking, if to be part of the Global Biodiversity Framework, you have to have indicators, then it's hard to have genetic diversity there because almost by definition, it's hard and expensive to monitor genetic diversity, but maybe not. This is the common perception of people when you talk about genetic diversity, that you have to do sampling, lab work, sequencing, and this is time-consuming and very expensive, out of the scope for the reality of many countries. Therefore, genetic diversity is almost not monitored globally. And although DNA-based monitoring will remain important for research and is something that I personally want to continue to be doing, we also recognize that for large-scale monitoring, we need easier and more affordable indicators. And that's where we, and when I say we, is a very large group of researchers, part of a coalition of conservation genetics that I will be talking about later. We designed the genetic diversity indicators as part of a global biodiversity framework, thinking in this reality. We started by asking at the population level 2 fundamental questions driving genetic diversity loss. Are distinct populations being maintained? And are those populations large enough to sustain genetic diversity? Based on this, we generated the proportion of populations maintained, which basically tells you if the different alleles that occur in a population are continued to exist because the population still exists, or if they have been lost. And then we have the NE indicator, which is the proportion of populations large enough to avoid losing genetic diversity by drift. The proportion of maintained indicator is relevant because distinct populations often hold different composition, different traits, behavior, physiology, different adaptations. And this is something that even non-experts can understand because populations living in different conditions often have different adaptations. The proportion of populations with an effective population size above 500 is more theoretical or more technical because it's based on the well-known fact on population genetics theory, which is that when effective population size starts to decrease, genetic diversity starts to get lost just by genetic drift. This is already also recognized in many, you might be aware of, in many forestry management, in agriculture and in fisheries. So to some point, managers already are aware of the importance of the population size. And it's important to highlight that we are focusing on the contemporary effective population size, which measures the rate of loss of genetic diversity within populations. And this is different to the census size because the census size are influenced by ecological aspects of a population, while the effective population size is based on the genetic level. And we know from decades of research that the effective population size usually is around 10% of the number of mature individuals. And of course, this varies by taxonomic group, but in general, 10% is a reasonable conservative estimate for very different taxonomic groups, including plants and all sorts of animals. But of course, there are exceptions. So the fact that we can have this ratio means that although we can estimate effective population size from genetic data using bioinformatic pipelines, we can also approximate effective population size based on the amount of mature or the number of mature individuals of a population, which is faster than performing DNA studies, and in many cases good enough for a quick monitoring. So based on this, we developed the indicators, and the effective population size indicator was adopted as headline indicator. This means that it's mandatory for parties to report on 2026 and going forward. Both indicators range from 0 to 1, where 0 is the worst case scenario and 1 the best case scenario. And I have been talking about we. We is the Coalition of Conservation Genetics, which is a very large group of researchers from all over the place. We developed the coalition as part of attending COP15, where we wanted to have a united message of the importance of genetic diversity. This allowed us to attend COP15 in Canada in 2022 and to present indicators and discuss them with practitioners and delegates from the parties of the CBD. Something else that we did at the time was to show that it was possible to estimate the indicators by actually doing it. So we ran this kind of like trial with 9 countries of people that literally raised their hand during a webinar in preparation to COP15. The effort was led by Jessica da Silva from Zambia in South Africa and from myself when I was based in Conavio in Mexico. And what we aimed to do so was to assess around 100 species by country and to show the preliminary data as part of COP15. The study is now published in Ecology Letters if you want to have a full read at the results. But here I'm just going to summarize that we got data from anywhere that had already published data on census size or effective population size. And this included not only scientific papers, but also species management reports, site inventories, even citizen science, or asking park rangers and local managers about the population sizes that they have. And I want you to think about if you are participating in projects that are generating this type of data, and if you have thought that this could be used for genetic monitoring. At the end, we were able to assess around 900 species and more than 5,000 populations on all sorts of taxonomic groups in all these different countries. How we did it was to first expend months and months and several Zoom calls, creating a shared methodology to assess the indicators in a reproducible and meaningful way. We developed a series of guidelines that we then made public. You can look at them with more detail later in order for others to be able to replicate or improve our methods. We had this web form where we capture data in a standardized way, asking specific questions for each of the populations in a reproducible way. This is an example of how our form looked. We developed different methods to define populations. More than develop, we recognize different methods to define populations, and we try to be as consistent as possible when reporting them. We have a decision tree for gathering population size data. We always prefer genetic estimates of NE, but when there is no genetic data, then we can use a number of individuals as a proxy. As an example of how our form looked when we were gathering effective population size data, it's like this. We have the point estimate, and we also have metadata on the genetic markers and the method that was used to estimate the indicator. When we have census data, we also try to account for the type of origin of the data. And importantly, in many cases, we didn't have the actual number of individuals for census data, but we were able to have just an estimate of whether it was more than 5,000 individuals, which will mean an effective population size of more than 500, applying the 10% ratio. This was particularly useful when we were talking with land managers or park rangers, where they won't feel confident enough to tell you, yes, there is 170 trees, but they are confident enough to tell you there is definitely less than 5,000 individuals or definitely more than 5,000 individuals. And in this way, we could recover more data to estimate an approximation of the effective population size at local levels. Just to give you an example of how data look at the end, this is a species from the US, this very nice flower that has very detailed counts at the population level in these different mountains because it's a protected species. Based on this, we could apply the 10% ratio to the population sizes to estimate effective population size. And we were also aware that no population had been lost, and therefore the indicator value is 1 for the proportion of maintained populations and around 0.4 for the populations above the effective population size of 500. This is another example from Mexico. This is a mountain juniper. Here we were able to estimate the proportion of maintained populations based on citizen science and herbario collections, because we noticed that there was a couple of mountains that had good data from the 50s or 60s, but then collections stopped in this region, and no citizen science observation had been made. And when you zoom in, you can see that these mountains became a mining site and had been basically destroyed. So it makes sense that these populations don't longer exist. Based on this, we estimate the proportion of maintained populations indicator. And based on asking park rangers for the number of individuals of these junipers in each of the different mountains, we were able to estimate the effective population size indicator. So repeat that for 900 species, and this is what we found at the global level. We found that the majority of species have an indicator value close to 0, which means that the majority of species have all the populations too small to retain genetic diversity. And this is true for both animals and plants. Then we have, the results from the proportion of maintained populations, and we found that the majority of species still maintain all of their populations. Something else interesting that we found is that although we were expecting the indicator value to be small in endangered species, we were not expecting to find it to be small in less concerned widespread species. However, this is the case for a good amount of species, which means that with the genetic diversity indicators, we could detect loss of genetic diversity that otherwise would be unnoticed because people don't tend to focus a lot on least concerned species. So finally, to close this talk, I want to show you how we can leverage other ongoing efforts and tools to monitor genetic diversity. First of all, you might be aware that the IUCN Red List has different workflows in workshops or desk-based approaches to estimate the Red List status of a species. And these workflows involve a lot of population data. Therefore, it's asking different questions with the same data. We could be more effective to estimate the genetic diversity indicators. So to test for this, we have been running a couple of examples. This is a workshop in Thailand that was happening to assess the risk status of a group of plant species with local botanists, and we were able to, at the same time, implement the genetic diversity indicators with very little extra effort. And this is something that we will be publishing soon, thanks to Thai colleagues, by the way. There is also the Genetic Diversity Scorecard, which is an effort that was undertook by the Scottish colleagues, Nature Scotland, and the Royal Botanic Garden of Edinburgh, and other colleagues. This scorecard asks several different questions addressing the main sources of loss of genetic diversity that species can have. And here, the genetic diversity indicators of populations maintained and effective population size are also included. And this is more detailed work that can be done with land managers at the local level. And we will be publishing an updated version of its scorecard soon. So keep in the loop. Finally, there is also this other initiative called Genes from Space. It's a team directed by some colleagues from the University of Zurich in Switzerland, who developed a method and a tool to monitor habitat size using Earth observation data. And in turn, this habitat size can be used to approximate the population maintained and the persistence of populations, as well as an approximation of their size. This is the link to the monitoring tool, which is still in a pilot stage, but I encourage you to have a look. And there is an ongoing project called GNOA, Genetic Nature Observation and Action. It's a cost action from the European Union funding that aims to achieve the conservation of genetic diversity across Europe. There's a lot of focus on assessing and using the indicators, but also it includes capacity building and engaging with stakeholders. And this is something that anyone from European institutions can join. So please have a look if you are interested. This is directed by Ankunta Fedorka. And with that, I would like to finish. Thank you very much, and I'm very happy to take questions.
Thank you very much, Alicia, for your presentation. I'm sure that there are a lot of comments and things for discussion. But as I said at the beginning, we will take now like 1 or 2 questions mainly to clarify anything. If you have any questions to clarify things, but otherwise we will keep the main questions for the discussion. Ophélie?
Yeah, just thank you very much for the presentation. I wanted to know how you deal with new populations, because if you had like a metapopulation dynamics, you lose some population, but there are also new population colonized. So how does it how is it taken into account in your metrics of proportion of maintained populations?
Thank you. This is a good question, and honestly a headache that we have. In the guidelines, we have a section where it's the best we can think of right now is to consider meta populations as the populations, as a unit to analyze. So what you will be tracking is the meta population, the distinct meta populations, instead of like the local dynamics happening. That's one possible solution, but this is something that we keep discussing. So if you have any better idea, it's more than welcome. I can put the link to the guidelines to that section where we are to the solution that we achieved for the purpose of the present study that I presented. But there might be other ways of dealing with it. It's a difficult situation. I recognize that.
Thank you very much. If there are no other clarifications for the moment, we can move on to the second speaker so that we are back on the schedule and on time. So now I will give the floor to Geir.
Thank you. Yes, I was invited to give a talk about salmon, which I guess is a bit not the organism that a lot of people of you work with. I have an impression that most of the people here are forest people, so I hope that I will talk about something that is interesting for you. So, but let's see. So, the Atlantic salmon is a fish that is born in the river, where it spends 1 to 3 years before it migrates out into the open ocean and have this long distant feed-in migration that may last several years. So 1 to 3 years is the typical time it takes for a salmon to grow at sea and get big and fat and go home to its natal river where it spawns and give birth to a new generation of salmon. It's distributed across the North Atlantic in a few thousand rivers. So each dot here is a river with salmon in it. We had started to monitor the total abundance of salmons in the early 70s, and it has really declined since then. So from approximately 9 million fish at sea, now we are down to a couple of million fish in the North Atlantic. So this has led to classification of the Atlantic salmon as near threatened on the IUCN Red List. Salmon are locally adapted to the river. They have this ability to finding back to the river where they're born. So we get this strong genetic differentiation among the different salmon populations in several different traits, like number of years they spend at sea. So some populations have a lot of old salmon in them, and some have younger salmon for the adults. Also, the number of years they spend in freshwater before they emigrate to sea differs a lot. The adult size and how well they grow. prime example of local adaptation is some large effect loci that have really strong differentiation among population compared to neutral markers. And these are associated with the number of years the salmon spends at sea. So some strong evidence of genetic differentiation and local adaptation in the wild Atlantic salmon. And then we have the farmed Atlantic salmon. So in the 70s, they started this breeding program in Norway, where they tried to select for a salmon that grew fast, had increased growth, avoid early maturation, better disease resistant, better flesh, nicer flesh color, and better fat content, to make a commercial product that was valuable. And this has been a very successful program, and they have really changed the salmon into a domesticated species, and now they're on their I think it's about 13 generations of artificial selection. And it's also been an economic a project that has is worth a lot of money. So now we have an enormous amount of farm salmon across the North Atlantic. So here you can see the different countries, and the blue dots are, one dot is approximately 100,000 fish. So the blue dots are the wild fish, and the orange dots are the farm fish for different countries. So in Norway we have about 400,000 wild salmon swimming back from the ocean each year to spawn in the river, but we have approximately 340 million farm salmon slaughtered every year. And Norway is the absolute biggest producer, but we have other big producers across the Atlantic also. So Scotland produces quite a lot of farm salmon. Canada has the largest wild population, so above 600,000 wild salmon, but still more farm salmon, even if it's not that big a producer, so about 8 million farm salmon. Of course, some of these are kept in open net pens, usually out at sea, and of course some of them escapes and swim into the wild and can find their way up in rivers and spawn together with the wild salmon. So I'm in a group of researchers that are interested in measuring how big this genetic effect is on the wild population. And my colleagues, and in particularly Sten Karlsson here at NINA, have developed a set of genetic markers that can be used to find out how much of your genetic relatedness is to farm salmon versus wild salmon. So we have this method for that uses structure to figure out where on the range between farm salmon genetics and wild salmon genetics a fish is. So we can measure if it lies in the middle, it's half of its ancestors are from farmed fish. And you can use different genetic markers for different types of wild fish. So in Norway, we have primarily two groups of genetically wild fish, one north in Norway and one for the rest of Norway. And we have recently updated the set of genetic markers also, so we try to develop these tools. But the main point is to be able to go into the wild, capture a fish, and estimate how much of its genetic ancestry comes from farmed salmon. And this has been done in several countries now with a similar methods. So in this plot, you see the distribution of all the salmon rivers in the northern Europe as dots. And you can also see where there is farm salmon production. So that's in this orange color that is along the whole Norwegian coast, but also in the Faroe Islands and 2 places in Iceland. And then we have the rivers where we have identified so-called genetic introgression from farm salmon. So we have found individuals that are hybrids between farm and wild salmon out in the wild. So in Norway, we have investigated almost 300 rivers, and in 178 of those, we find evidence of hybridization between farm and wild salmon. And you have this has also been done in Canada and in Iceland and in Scotland and even in Ireland, but those data are not available yet. So it's been a huge transatlantic effort into documenting this genetic effect on wild salmon populations. And the worry is, of course, that almost all salmon that is used is Norwegian, and it has been changed quite dramatically. So you end up by homogenizing the wild populations, particularly in countries outside or in other countries than Norway, where you have this effect of the Norwegian genetic background also from the farm salmon. And we have documented that this has a quite strong effect on the properties of the hybrids. So when you're halfway farm fish, then you have a different type of life history than if you are a pure wild fish. And the general tendency is increased pace of life. So this is a table that I found from the literature about pace of life. So the different traits that are associated with a short generation time, and typically I've highlighted where we see that where there is evidence for the genetic effect of farm salmon to induce a shorter life, early reproduction, higher growth rate, high dispersal, high aggressiveness. So it changes a number of these life history traits, behavioral traits, and physiology traits that is documented in the literature. So because of this worry that we are altering genetically our wild populations, the salmon got its own so-called its own royal decree from 2013 in Norway. So it's called the quality norm for wild Atlantic salmon to strengthen strengthen the possibilities of preserving the wild salmon in Norway. And it's two norms, one that is about the number of fish and harvest potential. So we want to have enough fish in our rivers so that people can harvest them. So a demographic goal of having high abundance of salmon in the rivers, but we also had this genetic goal where gene flow from farm salmon is one of the things that is measured. And to monitor whether how the different populations of Norway are, the status of them in comparison to these quality norms, we have a scientific committee, so the Norwegian Scientific Advisory Committee for Atlantic Salmon, where each population is evaluated for whether it has the total abundance in the population, whether that is high or low, and whether the genetic integrity of the population is maintained, and then with regards to introgression from farm salmon, and we have this genetic tools is been used on 250 rivers now. So we see that only only a third of the Norwegian rivers do not have any evidence for genetic introgression, and equally many has what we call the large genetic changes. That means that more than 10% of the genetic material in the river is from influence from farmed salmon. Let's see. But does all this scientific work have consequences? Yes, we have gotten new regulations for technical standards for fish farming installations. So they have rolled out rules that where there are where the pens where they keep the farm salmon needs to meet certain standards before they're allowed to use to mitigate the part of the SKP problem. And we have protected areas in Norway. So they have sketched out the most important fjords and rivers and made protected areas from though it's not allowed to have fish farming in those areas. So it has had practical consequences for management, all this scientific work to document the influence of farmed salmon, genetic influence of farmed salmon on wild salmon. In addition, the aquaculture industry has this program where they fish out farm salmon from the rivers, where they can they snorkel down rivers. So this is a person that is swimming in a river with a harpoon, trying to identify farm escapees. So you can see them you can have some so they have some morphological characteristics, so you can differentiate between them and wild fish and try to shoot the farm fish. Unfortunately, this program has not been very effective, but at least they are trying to get rid of the farm salmon, the farm escapees from rivers. And we do observe that the amount of farm salmon observed in the river declines over time. So this is the y-axis here is the proportion of escaped farm salmon observed in the river with 3 different methods. So this is in Norwegian, so it's not so important exactly what they are, but they measure this in 3 different ways, and at least from the early 2000s and until today, we luckily we see a decline in the proportion of farm salmon in the Norwegian rivers. So there is some effect of this legislations and increased awareness of the problem of having escaped farm salmon and its genetic influence on wild population in Norway. Yeah, and the last management implication of farm salmon is that we control all the brood fish that we use. So several rivers are stocked with local hatcheries. So they take out fish from the local river and they produce offspring which they put back into the river again. And when they use brood fish, they have to test it genetically, whether it's has genetic signatures from the farm salmon. Then they are not allowed to use it. Also, we have some rivers that we have in live gene banks because the environmental situation in a part of Norway is so bad that we fear that the local population of salmon will go extinct. So they decide to take this fish in and keep them alive in lab facilities. And then we also check these fish for genetic introgression from farm salmon before it goes into these live gene bank. So that's also an effect of all this work on documenting genetic introgression from farm fish. Yes, thank you. I hope that there is something to learn from the salmon story on other organisms also. And yeah, thanks for the attention. And if anyone has questions, then I'm happy to take them.
Thank you very much. Yes, we can speak the same genetic language, I think. So it was very interesting, and probably most of us are consuming salmon. So it was very nice to to listen to this talk. Any questions regarding clarification? Marimete.
Yeah,
And
thank
then
you very
François.
much, Geir. I'm just wondering, so what are the limits when you're talking about introgression? Are they then just pure wild salmon, or how do
Yeah.
you judge the
So
introgression limit?
we have 4 categories. So we have this no observed genetic changes, and then we have small genetic changes. And this is 1%. And also it's also partly a statistical significance test, whether you can say that you have something or not. So it's not, it's sort of a mixed definition. So they measure you have the point estimate of how much there is, but they also evaluate the uncertainty of that point estimate, whether it's in the no documented changes or smaller genetic changes. And then from moderate genetic changes, that's from small genetic changes is from 1% genetic introgression in the river to 4%. And moderate is from 4% to 10, and large are above 10. That's the values that I used. But of course, this is it can be discussed what is large and what is small. It's more so it's not from first principles, right? It's not like these values are easy to derive. You have to decide some values and argue for them, but it's not easy to find theoretical models that will tell you exactly what these values should be.
And you don't use any phenotypic characteristics along with the percentage of introgressed genes?
Not in this evaluation.
No.
What has done to be, there are some simulation models and theoretical models that have been used to study what level of introgression that would lead to demographic effects. But of course, it's based on a huge number of assumptions, and it's not so easy to, and you know, they also, the link between genetics and demography there's a lot of things happening between those two. So it's not an easy task to translate it into demography. It's easier to translate it into life history and morphological changes and these, but that is not sort of the managers are more interested in the demography than in the morphology of the fish.
Great, thank you. And François, you had your hands up.
Yes, thank you. I was just wondering, beyond the impact on the genetic integrity, is there any measure or evidence of the impact on the gene diversity of the wild gene pool, of the effect of this introgression that could happen by reducing the effective population size in the loss of wild gene diversity?
Yeah, that's an excellent question. So there is one relatively old study that shows that the FST, so this measure of among population variation in the genetic diversity, is reduced with the gene flow from farm salmon, and this is because the farm salmon has a relatively small genetic diversity. Right, so when you spread it over a large geographic area, you spread the same type of genes throughout all the populations. So there is one empirical study that shows this, but we should redo that studies with many more populations to really see this effect. But there is also a very good theoretical study that shows how one-way gene flow basically links gene flow and E. So if you have a system of populations with asymmetric gene flow between them, then you can have a really strong reduction in the total effective population size of the system. And basically with this one-dimensional gene flow like we have in salmon, the effective population size will almost entirely be decided by the effective population size of the farm salmon on the long horizon. And that means that there is an enormous reduction in the total effective population size in the affected populations. So it's it would be really cool to use that theory more in relation with the empirical data to see what type of effects we can expect in these populations here. So that's I can share that. I can share that theoretical paper if you guys are interested, because that's something that is not just for salmon. It can be used on anything. I can find that paper and share it in the chat if that's great. I'll do that.
Great, thank you very much. So let's move on now to our last speaker for today. So now I will give the floor to Ophélie.
Yes, let me share my screen. Okay, good. Can you see my slides?
Yes, we can see. Yes.
Good.
Thank you.
So it's a good transition because I will tell you about theoretical work this time during this talk. And I will try to tell you, so I will talk about some of my own work, but also the work of others. And I'll talk about evolutionary skew, so the link between genetics and demography, and how to model it, and what they taught us about the risk and benefit of assisted gene flow. So I will first start by thanking my collaborators on this different projects, Julien Fresson, Olivier Coteau, Adèle Ehrlichman, and Sally Otto, and diverse sources of funding. So I will start with a very concrete example, a plant, some endangered protected plant we've been working on in my team for a very long time. So Santora corambosa is a cliff specialist. It's found only in 6 populations with a very tiny range of less than 3 kilometers square in one small mountain near Narbonne in southern France. The life cycle of the plant is quite long-lived, but it flowers only once and dies after flowering. It's self-incompatible and has very low dispersal capacity, which means that once a population is extinct, there's very little chance that it will be recolonized again. We've been following the different 6 populations since 1994. This work was started by Isabelle Olivieri, for some of you might know her. Throughout the years, the demographic data we've collected suggests that all populations are on average declining, with acidic population growth rate that are below 1, with of course fluctuation between years, but a consistent decline and also an aggravating decline in recent years. And one cause of aggravation of this decline is climate change, which really accelerates the species demise. In particular, because strong heat waves killed the rosette stage, which is the most critical stage in the life cycle of the plant. And so again, demographic modeling suggests that in the future, if there's a continued increasing frequency of dry years and hot years in summer, the extinction probability of the whole species is will increase a lot, and there's 20 to 90% chance of extinction in 20 years for the species. So really high short-term risk of global extinction of these rare species. So because of that, and so this threat is just illustrative of threats that are concerning a lot of species due to climate change throughout the world. And so managers have asked for some intervention to try to buy time for the species. And so there was a program of population reinforcement that was implemented starting in 2018. And about 4,000 seeds were introduced in natural populations. Those seeds were produced in a garden where we multiplicated some plants from 5 of the 6 extant population of Xanthorrhoea corambosa, and were able to make the plant produce a lot of seeds. But those seeds were produced in the common garden without isolating the different populations, and so those populations in the wild are really genetically isolated, with very rare migration between the 6 populations and very large FST between them, about 30% or 25%. And here in the common garden, those plants from different populations could interbreed freely. And so there was an event of genetic admixture in the garden, and the seeds we reintroduced from in the natural populations corresponds to some kind of artificial gene flow event that we implemented by reinforcing the population, not only with more individuals, but also for individuals coming from an admixture between the existing populations. So this reinforcement had a minor demographic effect because we can show that we actually increase the number of flowering plants in the different population by 10%, which is not a lot given the very small size of those populations. But 10% is not insignificant in terms of from a genetic point of view. And importantly, because those individuals are coming from between population crosses, we might expect that we have introduced new genetic variation in the different extant population. So maybe the genetic variance for adaptive traits is higher due to this reinforcement population. And it could help the different population to adapt to climate change and cause them to be rescued. So I will actually already give away the conclusion. So the main question is, can this kind of type of assisted gene flow rescue a population from extinction? And these are going to be the conclusion of the talk. So I'm going to show that there's an optimal level of introduced genetic variation that maximizes the probability of evolutionary rescue. But that genetic variation for different life history traits does not have the same effect on rescue. So it's not only a question of the quantity of genetic variance, but genetic variance for what. And also I'm going to show that if you intervene late to rescue a population that is already very small when you try to rescue it, as is in the case for Xanthorrhoea, you can maybe save demographically the population, but most often it will be at the cost of losing all local genetic diversity. So why do we think that it might be a good idea to use gene flow to rescue a population? So there are indeed in the conservation genetics several examples of where either natural or human-assisted gene flow introducing individuals from foreign population was able to release inbreeding depression and consanguinity and result in positive demographic consequences in a number of enlarged populations. We also know more and more examples where adaptation to novel environmental conditions in different species is due to integration of genes that were coming potentially from different species or at least different populations. And there have been models. So the question is, if you have, we have seen this positive demographic effect. We know sometimes that some adaptation to some novel conditions or to stress more generally can come from genes from outside. But theoretically, already in the 90s, Komulkavitz and Holt formalized through simple models the under which condition indeed evolution could prevent extinction. So the scenario they considered was that of a population declining, just as the Xanthorrhoea corymbosa population is declining. And you can imagine if you have a simple population genetic model that you've got a resident allele with a negative population growth rate. So this, as a result of the population being mostly composed of individuals carrying this allele, the population is declining through time and will go extinct if no other genetic variation is introduced. And you introduce at low frequency a different allele, which is called a rescue allele, which has a positive growth rate. And this rescue allele will increase through time. And as the number of individuals carrying this allele is increasing, at some point, the population will start growing again because there's a majority of individuals carrying this allele rather than the resident allele. And you will see population numbers increase again. So this simple model is connecting the genetics through the change in allelic frequencies to population size. And these rescue models are typically illustrated by this kind of U-shaped curve in terms of population demography, where you have at some point a minimal population size in the course of adaptation. And if this minimal population size that is reached in the course of adaptation is too low, there's a high chance that despite the presence of an adaptive allele, a rescue allele in the population, the whole population would go extinct stochastically in this critical period of very low population size, and it will go extinct despite the fact that it was actually adapting to the new environment. So you can do the same kind of modeling by assuming not a simple population genetic model with two alleles, but assuming some trait with a quantitative trait with some initial standing genetic variants. And you can show in this case that there is a critical genetic variance that will allow evolutionary rescue and allow to avoid extinction. So again, if the initial genetic variance for the adaptive trait is high, you will have quickly evolution of positive growth rate, and the minimal population size reached will be high, and you will never be at high risk of extinction. If the genetic variance you introduce is too low, the population would ultimately adapt and grow again, but it will, in the meantime, go through very low population size, and below some threshold viable population size, the risk of extinction are really high. So there's a critical genetic variance that allows the population to stay above this threshold viable population size at all times during the course of adaptation. And this notion of a critical genetic variance was discussed by Gomulkowicz and Hull in a 2009 paper. So this is very interesting and theoretical. Can we use these models actually to try to predict anything about some real concrete cases such as the Xanthorrhoea corymbosa? So one difficulty is Xanthorrhoea corymbosa has a complex life cycle with individuals in stage seedlings, staying in the rosette stage for several years before growing to size that are sufficient to flower. And once they flower, they produce again new seedlings and die. And so the question we had more generally is how can we define a critical level of genetic variance for a species with such a complex life cycle? So one thing we did first is try to parameterize those simple quantitative genetics model with empirical demographic data. And so we used the demographic data we collected on Xanthorrhoea corymbosa for many years. So we had 20 years of demographic data for 6 populations. And we parameterized the statistical model, assuming that there was some latent phenotypic trait under stabilizing selection affecting different life history traits. So the different life history traits are maximized when the latent trait has some optimal value. And this fitness landscape is parameterized by using as a maximal value for each life history trait, the maximal value we observed in our demographic data, and estimating the current level of maladaptation of this theoretical latent trait has through the difference between the maximal observed value of some life history trait and the current observed value of this life history trait in the declining population of Xanthorrhoea corymbosa. So our model use a structured demographic model, but also underlying that the evolution within those stages of some latent phenotype with a Gaussian distribution, a constant genetic variance that is introduced at the beginning, and it also assumes fast demography compared to evolution. And so once we do that, we can simulate trajectories of adaptation in populations that initially declining with the same rate as Santhera corambosa. And we can look at the trajectory of population size and record the minimal population size during adaptation, depending on the level of genetic variance we have introduced in the population initially through gene flow, through assisted gene flow. And so here on this graph, I've shown the minimal population size as a function of the genetic variance that we introduced for the fecundity. And what you can see for the different population of Santhera corambosa, with a large population with very thick lines and a very small population with very thin lines. So if you define some viability threshold, you could use different viability threshold. You can define the critical minimal genetic variance above which the minimal population size is always above the viability threshold. And you can also see from those graphs that there's also a maximal genetic variance above which all the population are actually declining to extinction, because the genetic diversity in these populations is too large and creating too much genetic load and causing the population to decline, even so the mean phenotype in the population is at the optimum. So it suggests that rescue is possible only within a bounded range of genetic variance for life history traits. So you have to introduce enough variance to avoid extinction, but not too much. But what is more interesting is that indeed we know that climate change is affecting differently different life history traits with differential effect on seedling survival, rosette survival, and fecundity, for instance. And also we know that different life history traits make different contribution to population growth. So this different effect of variation in life history traits on population demography is measured by demographer, classically by a quantity that is called demographic elasticity, which is a standardized measure of an effect of perturbation in some life history traits on the population growth rate. And what we can see is that, for instance, in the life cycle of Santhera corambosa, disturbing the rosette survival has a much larger effect on the population growth rate than disturbing fecundity or seedling survival, and disturbing the flowering rate, the rate at which rosette become flowering plants, has a minor effect on the demography. And so the hypothesis we wanted to test is whether the critical genetic variance for rescue was lower for life history traits with a high elasticity, meaning that we need less genetic variation to rescue the population if this genetic variation is mostly for traits with a large impact on demography. And indeed, it's what we observed. So here you have the same kind of graph I showed you with the minimal population size throughout the course of adaptation for different genetic variants introduced for, in blue, rosette survival, in green, fecundity, and in yellow, seedling survival. And indeed, we consistently with our expectation, we see that the critical minimal genetic variance is lower for rosette survival, which is a trait with a higher elasticity and higher impact on demography, and is similar for net fecundity and seedling survival, who have similar elasticity and similar impact on demography, which is less than rosette survival. Interestingly as well, you don't see flowering rate on this graph, because for no value of genetic variation for flowering rate, we are able to save the population. So introducing genetic variation for this trait alone cannot save the population, because it has too little impact on the demography of the Santhera. We also see that the effect of genetic variation is different across different life history traits, because the maximal genetic variance that allows rescue is different across traits, but it does not vary as a function of elasticity in this example. It varies mostly due to the maximal value of the life history trait and how much you can improve the population growth rate when the trait is maximized, which is not necessarily linked to its impact per unit on the population growth rate. So the next step for us, for Centaurea, is trying to actually estimate this genetic variance and the covariance between adaptive traits. What I forgot to mention is that those results were assuming genetic variation for one trait at a time, and we also can generalize those results when there's genetic variation and covariation between like these three traits. And so that's what we're trying to do at the moment. Finally, I wanted also to explain that these models can also be used to understand the risk of assisted gene flow. So indeed, assisted gene flow or population reinforcement with foreign material can have positive demographic effect, but it could also have negative demographic effect, as we started to discuss with the salmon case. So it could be to demographic swamping, for instance, if hybrid offspring have a low fitness due to outbreeding depression. But also can lead, if there's too much introgression, to genomic swamping, so the loss of genetic characteristic of the target population due to not outbreeding depression but hybrid vega, so that progressively the material, the local genetic material, is replaced with the one that you have introduced to rescue the population. So again, there are examples of that, and there's a nice review that was published some time ago in Evolutionary Application that suggests actually that when you review the literature about hybridization and extinction, you find many more examples of genetic swamping than demographic swamping. So often, gene flow will not have direct negative demographic effect, but it will lead to the loss or the reduction of the local genetic diversity. So this was a work of that my PhD student Adele Ehrlichman did with Sally Otto during a PhD, and she worked on a mathematical model to try to understand what is the compromise between saving a population through adaptive integration and the loss of the local genetic diversity. So you can consider a genome in blue of a local genome, and you introduce immigrants that carry some adaptive allele at some locus B that allows the population to grow again. The population is declining, exactly in the same scenario I was presenting to you before. And so after the allele B, that is adaptive allele that's fixed in the population, you can look at what is the proportion of the genome where that was introgressed with the introduced material and where you lost the alleles that were the local alleles before the introgression. So if you want to rescue the population, you want to fix the adaptive allele. And so if the population goes extinct because you have not fixed the adaptive allele, you have lost all local genetic diversity anyway. So to understand what is a fraction of rescued local genome, first you need to estimate the probability to avoid demographic extinction. But once demographic extinction was avoided by the fixation of the adaptive allele at locus B, you can compute analytically what is the fraction of the local genome remaining conditional on demographic rescue. And so if you compute that, the compromise is that the probability to avoid demographic extinction is increasing with the number of introduced individuals carrying the adaptive alleles. So if the initial frequency of the adaptive allele is large, there's more chance that you would avoid extinction and adapt quickly and be rescued. But the fraction of local genome remaining once you are rescued decreases as the proportion of introduced individuals is high compared to the number of local individuals at the moment of the introduction. So there's a tension between the two. And so on this graph, I show you the result of Adele. And so the expected genetic rescue corresponds to this fraction of rescued local genome. And this is shown for a different number of invaders that are introduced initially in the population. And the different curves with different colors correspond to what was the size of the resident population when you did the introduction. So if the population size in yellow here is low, it means that you have waited a long time before doing the introduction because the resident population size is already very small. And this is shown for different fitness advantage of the adaptive alleles that you are trying to rescue the population with. So what all those graphs show is that there's actually an optimal number of introduced individuals that will maximize or minimize the loss of local genome, or maximize the fraction of local genome that is retained, as you see in most of those figures. And also what this figure shows is that this optimal number of introduced individuals is actually very low if the endangered population is already very small when you do the introduction. So that's a yellow or yellow curve. And also, but the loss, in this case, the loss of genetic diversity will be large in all cases. So you've got a small chance to rescue the population anyway. And if you rescue it, you cannot introduce less than one individual in the population to rescue the population. And the fraction of the genome that will be replaced by the introgress, by the immigrant genome, will be high in all cases. So this suggests that actually the temptation to do assisted gene flow as a last decision emergency situation is actually the worst situation you could imagine, because this is really the kind of condition where you would lose the local diversity. So if you have to do the assisted gene flow, basically you have to do it quite early if you don't want to lose, if you don't want to rescue the population at the loss of the local genetic diversity. And this is just to show you that actually this analytical result that Adele obtained are actually reproducing some results that some simulations were made before with a quantitative genetic model, also showing that if you look at the proportion of pre-adapted individuals introduced and the timing at which you introduce those individuals in a declining population after a step change in the environment, actually the optimal strategy is to introduce 10% in this example of immigrants, but actually even before the environment changes. So this is 0.0 here is the time point where they start to deteriorate the environment, and the most favorable condition for rescue is once the allele was actually already in the population before the environment changed. So just to conclude, I will repeat my conclusion. I showed you that there's actually an optimal level of introduced genetic variation for evolutionary rescue. That genetic variation for different life history traits does not have the same effect on rescue, and so different. So not only the quantity of genetic variation, but which are the life history traits and what are the demographic consequences of this genetic variation is really important. And that late intervention lead to the loss of local diversity, and often you have to choose between if you can save the population, but you will not save the genetic makeup of the population. And I want just to tell you as an anecdote that among the 6 populations of Santoraco Rambosa, the smallest population that we had at the moment, the only individuals remaining in this population that we have are actually individuals that we have introduced, and we indeed have lost the genetic diversity in there, not because of the introduction itself, but because all the few local individuals have gone extinct, and the introduction was too late to save the population and retain this genetic diversity. Thank you.
Thank you very much, Ophélie. We will take a couple of questions as before for clarifications, and then we will move on to the discussion part. So any questions for clarification? I've seen a hand, but then I lost it, or it was a clapping hand that. Yes, Aspalter Samuel.
Yes, hi, thank you for the presentation. At the very beginning, you showed this graph where you said that within 20 years, there's a 20 to 90% chance of extinction. But at the end, after 100 years, the population would have been extinct anyways. So do you also have expectations of how much you improved actually the likelihood in a similar comparison with the different drought scenarios? Because I was wondering, why would you try to save it if it's lost in 100 years anyways?
So you lose it anyway because all the populations are declining. So it's just that it takes some time to decline to extinction, but in the current situation, the population are declining, so ultimately you're going to lose them. So what I'm showing is that if you introduce genetic variation and the population adapt to the new climate, you might again get positive growth rate, and then you might avoid extinction. You might still go extinct stochastically if it takes too long to rebuild population numbers. But at least it's if you cannot re-evolve positive growth rates, ultimately you will go extinct anyway. Is it clearer like that, or is it? Did I misunderstand
I think
your question?
it's kind of clear. Maybe it's too much detail for a non-geneticist. So at the end, can you estimate the likelihood of extinction after you did the ideal introgression or the ideal assisted gene flow?
So we did not directly. Well, it depends. In some simulation that I didn't show, we can estimate what is the probability of not going extinct. But in most of the mathematical work I showed, you consider that if you never reach a very low population size, you're gonna avoid extinction. So when I'm talking about probability of rescue, I'm talking about probability of not going extinct, which is
sure.
guaranteed
maybe,
if you have no evolution.
Yeah. Okay. Thank you.
Then Geir?
Hello, thank you for a nice talk. The estimate of additive genetic variance, where you showed that if you had very little, it was hard to have a genetic rescue, and sort of intermediate, you had high. That was on the natural log scale, right? Just raw natural log scale, or the x-axis? Yeah.
It was let me check. It was the genetic variation for the log life history trait. And
exactly.
And it might be as well that there was a log scale as well. I don't remember, but it's also on a log scale as well.
Because most of those, most values, if I get the scale right, lie also published estimates. So for life history traits is about 0.02, or if I get the scale right, so most published estimate of additive genetic variant, or the median estimates of meta-analysis of additive genetic variance of life history traits lies on your very left part of that curve. If I understand your x-axis right, so the 100 is just insanely large, right?
Yeah. So the difficulty is it's a log genetic variance. So again, it depends on the scale. It's not heritability. Okay,
no, that I understand. It's evolvability. If you multiply it by 100, do you get evolvability in percentage then, or?
Yeah, but I agree with you that indeed we are mostly, I mean, in the context we are looking at, we are mostly concerned on what is going on the left part of the curve, which is the critical minimal genetic variance. But it's still interesting to see. So I don't think we have to worry too much about too much genetic load. But as you can see, depending on the life history traits, this maximal genetic variance is also for different value. And so for the traits that are really important for the demography, you really need less genetic variation to save the population, but also increasing too much the genetic variation is also for those traits. This is where it's most deleterious. So if you have genetic variation for many traits, also at the same time, you multiply those genetic loads. But indeed, in most of the case, what we are really concerned about is the left part of the curve. I mean, we're dealing with populations that are small and genetically depopulate, so it's not, we don't have to worry too much about the maximal genetic variation. And also, or something I need to mention is that this version of the model, those the maximal amount of genetic variation you can introduce before causing extinction is also underestimated, because what happens in simulation is that the genetic variance is not constant, and if you have a lot of load, part of it is purged, and so you can actually introduce more variance than this. So it's not what we are really concerned about.
Great, thank you very much. So just to keep the time, I will now give the floor to Francois, but I would just like to remind him that there are a few questions in the chat that they were not, we didn't have time to reply. So you can use them for the discussion with these questions.
Okay,
as well. Okay, thank you.
Yes, thank you. Can you hear me? Yeah. Okay.
Yes, we can hear you.
Okay, so thank you for these presentations, and I'd rather go to start with more general discussion on the topic, which is we had 3 talks on different aspects and different types of biological organisms. And the general topic is about the detection and management of genetic decline and indicators, measures, and thresholds. So we have probably different goals. For example, for the indicators, we have indicators for decision, when to act, and yes, and then we have indicators maybe for monitoring, so how is the process going on, and we have also indicators for reporting, which provides probably a more global view of the picture of what is happening globally on genetic decline for multiple organisms. So for decision monitoring and reporting, maybe in some cases we need more thresholds. For example, for decision, we have a measure which is effective population size, and the threshold is, for example, 500. So we need threshold for decision. For monitoring, we need probably measures. And then comes the issue of do we adapt the measures, indicators, and threshold to available data, and then the available data becomes a critical limitation, or do we have enough data and the critical lack that we have is in the measure, the applicable threshold, and so on. So putting on this on the table, I will ask maybe the 3 of you to give your thinking of what is the most urgent need, or what is really missing today, and yeah, is it part of thresholds for decision, or what is the critical need for research and development, for example? So, and please, this is just to start, but if you any general question, please put in the chat or raise your hand, and we will give the floor to anybody. So who among you 3 wants to start with? Yeah, Alicia?
Yeah, I think I can go first, just because I have been thinking a lot about that question in particular. Regarding what do we do with the threshold or the data, I think it has to be dynamic. We have to aim for the best possible science and then adjust to the data reality, but that also can help to improve the data reality, and then it becomes like a loop where we are moving forward to best-based, science-based decisions. So I know, I'm just going to say this plainly, I know that the genetic diversity indicators that we proposed and that were adopted at the EUF are not perfect. And in the research community, there can be a lot of discussion of whether the threshold is 500 or whether effective population size is really the best metric. The indicators are there, are in the global international policy. And that by itself, it's an achievement that is going to help move forward the type of data and the more research that is needed in the results. So that's what I mean. It has to be like a dynamic thing. And the other point I wanted to say regarding your question is that I think that we have to reach outside of the genetics community. There is a lot of people on more like ecological studies or even just taxonomy or natural protected areas management that have data that normally is not valued and is not systematized, like this approximate number of individuals. And I think that when you start presenting not the DNA data studies themselves, but the genetic principles or evolutionary logic, then people buy in and are able to provide information, and they wish to get more engaged. This is what happened with the example on the Thailand workshop of IUCN Red List workshop. This is people that normally do not work with genetics, but they were very excited about the idea of being able to incorporate their knowledge into something that aids genetics. I hope that answers at least part of your question. Thank you.
Yeah, it did not completely answer to what you think is the most urgent, but Ophélie.
No, yeah, it's just about the indicators. The question that I have is that indeed I agree with you that often we won't be able to genotype everything, and so it's good to have a demographic proxy for genetic diversity. But I was wondering whether actually those proxy, like the number of population or the population size, are not already indicators of demographic parameters that are also listed in what you should record in those convention, or just in terms of good health of the population. Is it something new that people were not doing already, for not with genetic diversity in mind, but just in terms of with probability of extinction or fraction of the population that are in good health in mind? It seems to be that there's some kind of convergence between those demographic indicators and proxy for genetic diversity, and whether it's worth to have something independent, or finally we're just proposing the same thing.
I can answer that very quickly. The data is there, but it's clumped at the species level. So the example is the IUCN red listing workshops. During the workshop itself, individual populations are discussed, but then the assessment is done at the species level, and what was in the populations is lost in the final report. So what is different is the attention at the population level, and we can spend a lot of time discussing what is a population, but that is the main difference. And I think it's key because evolution happens at the population level. And that way we can, and also local actions and management, right? So that's the main difference, I believe. And it's just the hook to then have more genetic studies like the ones that were discussed here.
Okay, thank you. And to answer your question, François, to me, it might be difficult in terms of indicator for broad-scale assessments such as CBD, but I think one aspect that is still a research challenge for me, at least, is trying to indeed connect more the question of like, I mean, if we want to set threshold in some concerning genetic diversity, because genetic diversity in itself is the object of conservation, we can decide of threshold as we can decide of threshold of anything. We want to conserve 30% of habitats or something like that, and we might want to preserve 80% of genetic diversity. That's some kind of compromise. But if we want to preserve genetic diversity because of its consequences on demography and processes and ecological processes, to me there's still a lot of work to do to link genetic diversity and those ecological processes, and to know what is the demographic impact of having less or high genetic diversity in terms of research. But I think this kind of data it's really hard to get, so it will be hard to deploy that as a general indicator. But I think in terms of research and in terms of strengthening what is the role of genetic diversity in terms of process or services, there's still a lot to explore for me.
Yes, Geir?
If you want my opinion too, I completely second that from Ophélie, that this link between genetic diversity and demography is a big knowledge gap. But my fear is that it's very context dependent also, right? So it might not be too many general rules there. And the other big knowledge gap is this link between plasticity and genetic variation. So probably it's easier for a population to be saved if it can alter itself, also in a changing environment. If you have a good adaptive plasticity, so you could just sort of alter the way you behave or alter your phenotype in accordance to it, it's a higher probability of not going extinct than if you have to have genetic evolution to follow the optimum. But this link between the two is also a understudied point, but I'm not sure if it will help us in finding very good thresholds either. But in terms of understanding when population go extinct and when they can cope with an environmental change, I think that is an important research field.
Okay, there is a question in the chat. So Agathe Lévesque, are you here? Maybe you can directly. Are you here? I can read the question. Thank you very much for these excellent presentations. You have highlighted various aspects of genetic decline. I wonder, when we think about genetic decline, especially when we want to understand how fast and how strong it is, how might we define an appropriate baseline? I realize that establishing such a baseline is not straightforward, but without it, how can we truly quantify or compare the ongoing genetic decline among species? So Agathe Lévesque, don't hesitate to take the floor if you are here. But who wants to answer to this question?
Can you repeat it? I was trying to read the chat, but I think we don't have access.
By the way, can you read in the chat the question?
No,
or not?
we don't see the chat. It's
you don't see the chat. Okay.
I think. Yeah.
So the question is, I wonder, when we think about genetic decline, and especially when we want to understand how fast and how strong it is, how might we define an appropriate baseline? I realize that establishing such a baseline is not straightforward, but without it, how can we truly quantify or compare the ongoing genetic decline among species? So the question is about the baseline.
So maybe I can say something briefly. The baseline as in the year when you start to monitor. I don't know if that's a question. It's also a tricky one and is country dependent because the biodiversity loss started at different moments in time in different countries and also stop relatively in different countries too. So something that, and there is, at least for genetic diversity and the effective population size, there is this lag between genetics and demographic changes. So something that we encourage to do is to focus on effective population size of the current existent community as a way of looking forward to the diversity that will remain available in the future, and look at demographic changes of as many generations as possible to have a better idea on how much diversity has been lost and therefore is currently available. Linked to whenever the species started to have the biggest change in demography.
Okay, thank you. So Ophélie, you wanted to react on that?
Yeah, so I don't know, it's a bit, but I know that there's some papers that are discussing the idea of dark biodiversity. So they try to measure what kind of diversity we should find in some habitat, for instance. So it's a species diversity in this case, based on the regional pool of species and based on, you know, trying to associate different habitats with different diversity level. And so you're trying to evaluate what is the missing diversity that you don't see in some habitats that you should see given some given this habitat. And I was wondering whether we could do the same for genetic diversity and say, okay, given the life history traits of that species or related species, or given this kind of habitat, what kind of diversity we would expect in that species, and say what is the missing diversity that we don't have because of human impact. That would be a different way to you know, establish a baseline, because this idea of dark biodiversity it was trying to you know, take into account the shifting baseline or this kind of thing, like trying to have some expectation about diversity that would not be already completely arbitrary, as Alicia mentioned, because degradation has started at different time or stuff like that. Maybe it's not that simple. It's already not very simple for species diversity, but maybe there could be ideas like that for genetic diversity as well. Then we could develop research on that.
Okay, so again, thanks you for your answers. So I don't know, Geir, can people take the mic, or should I read the questions in that are in the chat? Eric, can you?
Yeah, thank you for very interesting presentations. So I put in the chat that I mean, we have a lot of focus on effective population numbers and this loss of diversity in these declining populations. But with trees, for many of the species, fortunately, they have life history that actually keep up population sizes because of pollen flow, wind pollinated, and or their large distributions. But then also they have the problem of having long generation times, and that actually could lead to fragmentation, could lead that they have a problem in adapting to climate change, for example. So my question is actually also what to what extent we should take into account whether these local populations have this sufficient standing variation, or we need to enrich them with something. So it actually, I think that's closely related to Ophélie's presentation. This idea that we should find out whether these declining populations are missing something that we should enrich in a targeted way, if we can do that. So, for example, if we have declining populations because of increasing drought events in fagus in beach populations, which have probably high population sizes per se, then we maybe should enrich some of these populations with genes which are adapted to more drought tolerance. So this is more like, whether we have the focus more on diversity per se, or, which kind of diversity, and can we really qualify how to enrich things? It's like, if you can follow my question or comment.
Yes, Ophélie?
Yeah, maybe I can answer. So yeah, indeed, this is something that's discussed a lot. Call it targeted gene flow, acid gene flow, often is, or trying to not increase genetic variation in a blind manner, but increase genetic variation that we think is pre-adapted to or is providing an adaptation. So indeed, in some situation, I think it could be a good strategy. It's not completely clear for me quantitatively how much you should increase the frequency of those adaptive variants to make really a difference in demography again. This is something we discuss a lot from a purely genomic or genetic point of view, but what is the demographic impact of changing the frequency of those alleles? And there are some simulation results that already suggest that if the population is already large and quite diverse, it's not so clear that it makes a large difference in terms of timing of adaptation or benefit in terms of service. But and also for me, the main issue with this is that it supposes that we really know what would be adaptive in the future. And so in some cases, for instance, with disease, where you've got a single source of stress that has a huge impact on the population, it's quite clear, and it's already used in conservation biology in trees, but also in animals, to try to increase resistance, for instance, to a disease. But in many cases, we don't really know exactly what would be the climate, exactly what would be the genes that would confer a good adaptation. And so there's a risk, as I was explaining, that if you increase one specific genetic variant, you will lose the genetic diversity in the population, especially if this variant is adaptive, and you might lose the capacity to adapt to other things that you have not foreseen or that might happen in the future. So I think it's an important question, but it's a tricky one, and I think we should be quite cautious when we introduce this novel genetic variation that we are not homogenizing and losing genetic diversity, and this is not something that is discussed enough in this assisted gene flow literature, I think.
And maybe, yeah, just to jump on this discussion and before giving you the floor, Geir, the tricky part is that, as you showed, Ophélie, it can be sometimes too late to intervene because, as you showed from your last example, but anticipate too much can be too much risk also. So maybe that means there is a window of decision, not a threshold, but a window, not too late, not too much anticipated, to avoid this risk.
Actually, what I showed is that anticipating, well, in terms of if you introduce a small quantity of genetic variation in a population that is quite healthy, the risk is very small.
Yes.
Because it won't swamp the genetic diversity. So so anticipating by replacing completely, the genotype, of course, is not good. But if you increase genetic diversity early on by just introducing some alleles that are new at low frequency, the risks are much less than if you do that too late.
No, it's a balance between over action or so. Geir, maybe.
Thank you. I think it's maybe at least one place where we know that it has been successful, and that's when you have a lot of inbreeding in the local populations, because then you know that putting in new individuals can have some real beneficial effects. Otherwise, I completely agree that it's a high risk. And the other one is so if you need to put in new individuals, it's off if you know. If you have an idea of what type of genetic variants that sort of belong to that area, you are much higher. I think you are more likely to be successful. For example, since I guess you are Danish, Erik. So they reintroduced salmon on the west coast of Denmark, and they tried with different type of fish, but then they found some that was sort of had the original genetics, and then they were successful. So it didn't work with the Scottish fish, for example. So if you have to do it, then to try to figure out what was the original genetics of these population and try to bring back some of that. In Norway, we have gene banks for salmon where we know we take good care of the original genetics to put it back out again in the hope of that the environment is going to be good at the point. And I think that's to just take put in a random genetic variation in a wild species is quite risky.
Alicia?
I just wanted to complement all these great answers by highlighting that all this is true, but when it comes to reforestation or restoration projects that include just adding a lot of new individuals, then that might be a different situation. And this happens not necessarily taking into consideration all these genetic implications, but mostly on what is feasible from the logistics way of producing seeds or plants in large quantities. But it's a big responsibility because due to the long life of these trees, decisions that we do now are going to last there a lot of time, as opposed to what happens with agriculture, where if you try a crop and it doesn't work and it died next year, doesn't matter because you can try again. But with forests and with forestry and with restoration, it's long-term decisions. So it's worth convincing stakeholders to invest more time thinking, what are we going to do?
Okay, thank you. Are there other questions? If not, we have some to be kind of not provocative, but oh yes, Jonathan, maybe. I have.
Hello, François. Thank you. Hello. I hope you can hear me okay. Just expanding on Eric's question and people's answers in terms of introducing individuals into a declining population. We're spending a lot of focus looking at the declining population, but not about the donor source as well. And I wondered whether Ophélie highlighted that there could be lots of different factors we're trying to counter and could be causing problems for the population. But when it comes to climate change, if we are focused on warming being a stressor for the flowering of a species, given in the example, then we know we're looking for populations that can tolerate that type of warming effect. And our donor population are probably, through climate change, going to be the ones that are even more stressed than the receiver population that we're trying to diversify. So rather than just looking at the receiver population's decline, is one of our triggers for intervening where the donations are going to come from and whether we should be proactive in that sense too. So we're introducing a very small proportion in the receiver population early on, but this might also be driven by the availability of material from a potential donor population, which might be in much more severe decline.
Ophélie?
Yeah, I really agree with that. And there are, again, modeling, especially in corals, suggesting that if you don't take into account adaptation and genetic diversity, for instance, there's a strong temptation when you protect different populations to protect populations that are in kind of climatic refugia, that are a bit cooler than in the cooler part of the range, because you expect the population there to persist even in warmer climate, because there will be like climatic refugia. And actually, if you take into account the necessity to maintain genetic diversity and maintain adaptation to warmer temperature, you need to spend time protecting also the population where we tend to think that those are lost and those are losers because they are, outside, they are going to be outside of the niche, or they are in the warmer part of the range. So there's a difficult tension here. Like if we, and potentially I'm a bit worried that there's some, strategy of prioritization of where we should put natural reserve or natural protected areas, trying to identify those areas of the range where you remain the climate you adapted to, but by stopping putting efforts in the warmer part of the range, we might lose very interesting genetic diversity. So I think it's something related to a question that we need to worry about, and related to what Alicia also suggested, that I think it's not only the total number of population, but also what kind of climatic or phenotypic diversity we're trying to maintain in the species to maintain their evolutionary potential.
Eric?
Yeah, thank you for the answers. And I think maybe this is a very good link for the discussion to continue tomorrow. Again, with Geir, with your results discussion from Norway. I mean, obviously it's more safe to introduce what we all know is already locally adapted, but just to be a bit provocative, to say in Norway, if you are facing 3 or 4 degrees increased temperature and climate change, will your local gene pool of your local species be able to cope with that? Okay, or will you need to rely on populations which are adapted to different climate conditions? But I think this is actually, I think it was good. Thank you for your answers to my questions, and I think this is a part of the discussion for tomorrow. Also, a bit about how we can actually, what we actually do in real life. As François says, we should not be too late, because then we introduce a lot of genetic drift, and also a lot of hijacking of genes. But if we do it, so we should. But if we do it too early, maybe we don't know what we should do. Okay. So just thank you for the answers.
Maybe there is any other point. Otherwise, yeah, to be a bit provocative, or well, in trees, most of trees have high genetic diversity, but some trees have very low genetic diversity and they behave very well. And this is one point. So why bother about genetic diversity if some species can do so? That means probably species with high genetic diversity cannot support to have a decrease in genetic diversity, and is still high for them. High values of genetic diversity can be a problem because it's a strong decrease, while for others, so this is a point. And there is also the case of tree species that do not make populations. Actually, there was the case mentioned by Ophélie about metapopulation, and what Alicia also answered, but there are also cases of trees that behave like dispersed individuals and dispersed in the landscape at very long distance one from the other, and they are like this for a long time. So the thinking in terms of population is also challenging in those cases. So my question behind is, how far can these indicators, measures, and so on be sufficiently generic? Or, okay, for monitoring at global scale, I understand we can have something generic, but for specific decision, how far can we have generic thresholds, or should it, in any case, are there general rules always true, or does it need to be specifically developed in each case? It's, yeah, Alicia or Geir?
You're first. Well.
Okay, no, go on, Alicia.
No, you first. Yeah.
No, I was just going to speculate further, so I don't really have any good answers to it. But I think it's a very good question, and I think that this is always, you know, if all metrics have some strength and some weaknesses, right? And then you can always find specific cases where it's something else, but that doesn't mean that the metrics are not useful for anything. But of course, so some plant populations have had a lot of purging, so they can handle genetic loads to a very different level than or inbreeding, so that they don't get that high genetic load from inbreeding compared to outbred species, which cannot handle it. So of course there are situations where this is really different.
My question behind was, is it really the level of genetic diversity that matters, or is it the trend in genetic diversity that matters? I mean, the decrease rather than the value of diversity, this kind of question. Actually, effective population size is the measure of decrease rather than a measure of diversity, maybe. So, Alicia?
I also don't know how to answer the question fully. But I think that we should aim for something that is general enough and then attend the specific cases when they arise in a particular setting. And in many situations, it's obvious when it's going to be a specific case. Or there are some generalities like species that have a lot of asexual or clonal reproduction, for instance. But I do believe that we should aim to have monitoring of genetic diversity just as a general proxy, at least, in as many species or ecosystems as possible. And I think that we should be careful when we engage with non-scientists, that if we start discussing a lot or too much the details of whether it is possible to actually measure genetic diversity at all, it could be dangerous in the sense that we might end up not monitoring or not considering genetic diversity at all. So we have to just keep in mind that against what we are comparing. Are we comparing against the gold standard of having SNP data of hundreds of individuals by species, or are we comparing against not including genetic diversity in the decisions at all? So when I have to choose between like, okay, a proxy that works, that tells me something information, or not doing anything at all, then I just go for the proxy because I think it's better than just not accounting for genetic diversity at all. And this is especially true outside of the European context. I work at Kew, but I am originally from Mexico, and most of my projects are there in Latin America. And this is often the case. So when you have to choose between these two options, then proxy data and imperfect metrics are better than no genetic considerations at all.
Okay, thank you. And last answer from Ophélie, and I think now after time is over. So Ophélie.
Yeah, I also want to point out the fact that indeed there are populations with very little genetic diversity for neutral markers, maybe as well, that are doing well. It does not necessarily mean that they have low genetic diversity for everything. And also genetic diversity, I mean, it's not only inbreeding and stuff like that, it's the potential to respond to an environment that we cannot predict and that will be changing. And we know that maybe they're doing well now, but we also know that their environment is going to change. And if we want to protect diversity and the capacity to evolve and diversify and everything, we need to protect diversity. So it's not, I don't think the fact that there are populations that are genetically depopulated and doing well is a good argument to not try when we can to protect the diversity. It doesn't mean that we need to necessarily, do gene flow in this population at any price. But we know that something is not normal in the fact that they are very low genetic diversity in terms of long-term persistence.
Yeah, my question was purely provocative, to be prepared to have arguments to those people saying, well, why do you bother? Right. So thank you again. And I think the discussion went through science to pragmatism, and this was the objective of the seminar also. So thank you very much. So Anna Maria, sorry for exceeding the time.
No, that's fine. It was a very vivid discussion. So I would like to thank very much our speakers today, Alicia, Geir, and Ophélie, for being here and for giving these interesting presentations and for this very nice discussion that we had afterwards. So we will be expecting all of you tomorrow. Tomorrow it's a little bit earlier. It's at 11 Central European time. So we will have our second webinar, our second day of this webinar, and we are waiting for all of you tomorrow as well. Thank you very much and have a great day. Bye, everyone.
Bye-bye. Thank you.